So this is an interesting seminar because we are co-sponsoring this. This is typically the division of biostatistics and bioinformatics time slot, but SD reached out and we decided to co-host this seminar along with the CAPS methods core, which is probably why you see a larger number of participants than we actually usually have in most of our seminars. So the title of today's talk is Evaluating Benefit Risk, Handling Missing Data and a Universal Sample Size Formula for Clinical Trials. And we're fortunate to be joined by two speakers. The first is Jeetu Ganju, who's an independent consultant working mostly with small pharma and biotech companies. He has over 20 years of industry experience, both in pharmaceuticals and in biotech. And prior to his role as an independent consultant, he headed the biometrics department at Global Blood Therapeutics. An interesting other fact is that he is a board member of Room to Read, a nonprofit for literacy and gender equality. So that's always nice when people do stuff outside of statistics. Our second speaker is Ron Yu, who's a senior director of biostatistics and the head of Center for Statistical Excellence at Gilead Sciences. There, his group provides statistical consulting and training to two other project statisticians and non-statisticians, and he received his PhD from Stanford in 2005. So I think it's really fortunate when we get to have speakers from industry come to join us. So I'm looking forward to hearing what you all have to say today. Welcome.
Aaron, thank you for the introduction. And can I just check, do we have until 4 o'clock or can we go a bit beyond that?
So our time slot is typically up until 4. So okay, if you can wrap up around then, that would be great. But I also understand that you might have a little more material. So some people may need to exit, but the majority will be here till 4.
Okay, no worries. Thank you. And thank you for the introduction. And as Aaron just mentioned, the title is about 3 different topics. One is we will propose a metric for the evaluation of benefit risk, a joint assessment of benefit and risk. The second topic is we'll offer a non-parametric solution to the missing data problem. And I'll cover these first 2 topics. And Ron will speak on the universal sample size, a non-parametric universal sample size formula for parallel group trials. And it's universal in the sense that it doesn't depend upon the type of endpoint or whether the trial is of fixed or variable length. This last topic is joint work with Lou Tian of Stanford University. And these are big topics that we are going to be talking about, so we will cover this at a high level. And please ask questions, but if you feel that any question you haven't had a chance to ask, by all means, please reach out to us by email. One thing that these 3 topics have in common is that they are built on the same underlying idea. And the main idea is this, and I'm going to basically get to the main point right away. So just to fix things, let's just say we have 2 endpoints that we want to combine in a hierarchical way. And let's say the 2 endpoints are time to death and frequency of hospitalizations. And let's just say we agree that death is a more important endpoint than frequency of hospitalizations. So the approach is to compare pairs of patient data on the most important endpoint. And with this example, you would compare them on time to death. And then there are 3 possible outcomes when you compare pairs of patient observations, that whether patient A did better than patient B, patient A did worse than patient B, or whether the comparison was indeterminate, in which case we would call it a tie. And they would be assigned scores of +1, -1, or 0. Now, this is basically the idea of Mann and Whitney going back to 1947, and they did this in the context of a single endpoint. The idea was then taken further to include multiple endpoints by Finkelstein and Schoenfeld in their 1999 Statistics in Medicine publication. And what they said was, when you arrange the endpoints hierarchically, and if the endpoint, say with the example I gave, is a tie, say both patients are alive, then you would compare them on the next most important endpoint, which in the example would be frequency of hospitalizations. And the comparisons would occur at the minimum of their follow-up times. And the metric that we are going to be looking at in this presentation, I'm going to call that theta hat. It's the ratio of these two probabilities. So the numerator is the probability that patient A, a randomly selected patient from group 1, did better than patient B, a randomly selected patient from group 2. And the denominator is the estimated probability that patient A did worse than patient 2. So let's just take a couple of examples. All right, so that same endpoint 1 is time to death, endpoint 2 is frequency of hospitalizations. So we have patient A with 2 hospitalizations, but the patient subsequently dies. Patient B has 1 hospitalization and then also dies. And time is shown on the horizontal axis. And you see that patient B died before patient A. And because these are arranged hierarchically, you start the comparison on death. And if you had to figure out who did better, patient A lived longer than patient B, and we would say patient A did better. All right. Let's look at another example. It's the same setup, except that both patients are alive. So when you compare them on that first endpoint of death, it's a tied comparison. So then you would then make the comparison on frequency of hospitalizations. And here we would see that patient B, because patient B had 1 hospitalization compared to patient A who had 2 hospitalizations, patient B did better than patient A. All right, so it is this idea that we will take forward into proposing a metric for a joint assessment of benefit and risk. So the current approach is a subjective assessment of deciding whether benefit exceeds risk. Now, benefit is assessed formally through efficacy endpoints. We calculate p-values, confidence intervals, and so on. And so this is similar to what we do, for example, in criminal trials, where we say it's better for 10 guilty people to go free than for 1 innocent person to be wrongly convicted. So we have a framework for efficacy. For risk, it's done through the subjective evaluation of adverse events. And then when we have to combine the two, that evaluation is also subjective. And a question is, why is that? Why don't we have a framework for the evaluation of benefit and risk? And in this talk, we'll make a suggestion for how this could be done. I'll make a quick side note that this term benefit risk is not symmetric. Benefit refers to the gain that will be accrued. This is the benefit the patient will experience, and risk is harm that a patient may experience. And a neutral term would be benefit harm. But I'll continue in this presentation to use the term benefit risk. All right, so in the next few slides, I'll just walk through how we typically present data coming out of clinical trials. This is not going to be new to anybody, but I just wanted to set this up to then make a proposal for how this joint assessment of benefit risk would look in the context of a late-stage clinical study. So this is an example from idiopathic pulmonary fibrosis of an approved drug. It's nintedanib, or it goes by the name of Ofev. These are 2 phase 3 studies, Impulses 1, Impulses 2. This is a graph for the primary efficacy endpoint, which is the annual rate of decline in forced vital capacity. It's a measure of lung function. And the graph for Impulses 2 looks very similar. And so as you can see, it's presented with p-values, the point estimate, the confidence intervals, and so on. And then we have some secondary endpoints as well, and they could be alpha controlled. In this case, this key secondary endpoint is time to first acute exacerbation. On the left, you don't see much of a separation between treatment groups. On the right, you see that nintedanib did better than placebo. And then there are some additional secondary endpoints: % predicted of FVC, survival. The mortality rate in this 52-week study was on the low side, so one would say it was not adequately powered. If there was a real difference between groups, for the active arm, the rate was 5.5%. For placebo, the mortality rate was 7.8%. And so this is sort of how efficacy data get presented. And then when we come to presentation of safety data. This is pulled out from the US package insert. You will see there's some text around adverse events, something about myocardial infarction in this example, pneumonia. There's a paragraph on adverse events leading to study drug discontinuation, and so on. And then it's also summarized in a table, and I've pulled a portion of the table from the package insert over here, just to illustrate what is typically included. Now, it's uncommon to include multiple occurrences of adverse events. And just to focus on a couple of these events, diarrhea, for example, 62% of patients on the active arm had diarrhea, and 18% of patients on placebo did. With nausea as well, there is a difference between groups. It's 24% and 7%. And so then one has to sort of, review these data in a subjective way, and then sort of you have to review the totality of risk and benefit and make a decision, some judgment about whether benefit exceeded risk, but we don't quantify it, and there is no, clear pre-specified framework for that evaluation. So what could be done? And here is a recommendation. Before we break the blind for a study, we specify important efficacy and safety endpoints, and we arrange them by priority. So taking the IPF example, one possible ordering would be death is the most important endpoint, followed by acute exacerbations, and then something that I'm calling SAE1, serious adverse events of one kind, that would be less important than acute exacerbations, but more important than forced vital capacity, followed by forced vital capacity, and then SAE2, these are adverse events of another kind that are less important than forced vital capacity. And this arrangement allows for including multiple occurrences of, say, acute exacerbations, multiple occurrences of, say, diarrhea, and so on. All right. And the idea is to first calculate theta hat, the ratio of probabilities, and its confidence interval for the binary version of the primary endpoint. You might recall that the endpoint was forced vital capacity. It was a continuous endpoint. And the reason for making that a binary endpoint will become clear in a moment. And then the next step would be to calculate theta hat and the confidence interval in a sequential way. So this is, what the analysis would look like. And again, just to fix things, it's just easier to see this through an example. Let's say there are 100 patients in group 1, 50 patients in group 2. There will be a total of 5,000 pairwise comparisons. And this probability, the estimated probability that A did better than B, is the number of times that a patient in group 1 did better than a patient in group 2, divided by 5,000. And in a similar way, one would calculate the estimated probability that A did worse than B. And theta hat is simply the ratio of these probabilities. And here is the formula for the 95% confidence interval. It involves the variance of theta hat. And the variance of theta hat is shown in the bottom left. It involves the sample size. It involves the allocation ratio, which we would know through the design of the study. But it also involves the proportion of these paired comparisons that result in a tie. And as the proportion of tie increases, the variance increases. Now, this is an approximation, and Ron is going to give a more precise variance formula. All right, and just to briefly mention some of the relevant literature on this, I already talked about, Mann and Whitney's paper and Finkelstein and Schoenfeld's paper. And Finkelstein and Schoenfeld, by the way, didn't propose a metric. So their method basically gave a Z statistic from which one got the p-values. Boyce actually proposed taking the difference of these two probabilities. But again, there was no variance formula that was provided, and reference was made to the permutation approach for drawing a conclusion. Then Pocock and co-authors proposed taking the ratio of these two probabilities, and Pocock and company called it the win ratio. It has gotten a lot of attention since its publication. And they said that this variance formula is quite complicated, which is why they recommended the bootstrap procedure. And what Ron and I have is this formula for the variance shown on the left. And I'll get to that in a moment in terms of how it works in real life. I also wanted to quickly mention that this notion of combining efficacy and safety endpoints into a single metric was also proposed by Evans and Fullman. All right, so how would this formula look like in real life? So we tested this against 10 different data sets. Most of these are cardiovascular clinical trials. And for many of these publications, they mentioned that the bootstrap procedure was used to calculate the confidence interval, which obviously requires patient level data. And they also provided, and for many of these publications, Pocock was one of the authors. And we also used our formula, which just required summary level data. So in addition to the sample size and the allocation ratio, we needed to know what theta hat was and the proportion of ties. Just given that information, we could calculate the 95% confidence interval. And as you can see, there is a close match between the formula and what has been reported in the literature. Now, we know that this formula works well with endpoints that are positively correlated. Benefit risk assessment can involve endpoints that are not positively correlated, and we haven't investigated how well that formula would work in that instance. But again, as I mentioned earlier, Ron is going to speak to a more precise version of the variance of theta hat. So how would we use this for benefit risk assessment? And here is a made up example. So let's say we make forced vital capacity at week 52 a binary endpoint, and we could make it a binary endpoint by saying that the week 52 value is within 5% of baseline. So it's yes, no, whether it's within 5% or not. And let's just say theta hat, the ratio of those two probabilities is 1.5, suggesting benefit. And here's the confidence interval for that. Now, we had a hierarchical arrangement of the endpoints a couple of slides ago. So death was the most important endpoint. So let's just say theta hat is 1.03. There's the point estimate is in the right direction, but obviously the confidence intervals are too wide. Then you would add to that endpoint acute exacerbation. So acute exacerbation now is a lower tier endpoint. It kicks in only if the comparison on death is a tie, when you compare pairs of patient data. And we see that theta hat increases, suggesting additional benefit. Then you can add to that list SAE1, and again, theta hat goes down because, for example, in the example that I looked at of that approved drug, there was a higher rate of nausea and some other adverse events. And then you would add FVC, the primary efficacy endpoint, theta hat goes up. Then you add SAE2, theta hat goes down again. So this is one way by which one can get a joint assessment of benefit and risk. All right. So now I'll move on to the next topic, which is handling of missing data. And it's in effect the same idea. So let me first start off with what the convention is. The convention is to pre-specify one method, which is then backed up by several sensitivity analyses. Now, when we have different methods of analysis applied to the same data set, the results will vary. And again, we don't have a framework for figuring out what result should be considered to be more credible. There is a press release that comes out from the company. There is a publication in a journal, which involves people from academia. There is also the US prescribing information as well. And they may not have exactly the same information. And what is unclear right now is that, which result should be judged to be the most credible. I'll set that issue aside and get to what the main topic is ahead of us, dealing with missing data. So this is an example from a cardiovascular clinical trial called ATTRACT. The endpoint here is 6-minute walk test. It's the distance walked by a patient over 6 minutes. For this trial, a 6-minute walk test was a secondary endpoint. The missing data rate at month 30, so this was a 30-month study, all patients were followed for that same length of time. The missing data rate was very high. Only 51% of patients who were randomized provided data, 6-minute walk test data at month 30. MMRM, mixed model repeated measures, was pre-specified as the primary method of analysis, and then there were pattern mixture models for sensitivity. Now, the primary endpoint for ATTRACT was a hierarchical combination of mortality and frequency of cardiovascular hospitalizations. And as you can see in the study, there's a benefit on mortality, and also there's a benefit on the cardiovascular hospitalization time to first event endpoint. And they have contributed to missing 6-minute walk test data. And the reason I have at the bottom missing in quotation marks, the second missing in quotation marks, is because some people might say that if a patient does not survive to month 30, the data are not missing. The person, it does not even exist to begin with. And so this part I'm going to set aside in terms of how we deal with the missing data problem. All right, so this is now a table that is pulled out from FDA's statistical review. The main analysis refers to MMRM, and you can see the treatment effect is about 76 meters. The pattern mixture analysis resulted in a treatment effect of about 62 meters. So different methods of analysis will give you different answers. It shouldn't be too surprising. And this is what I'm calling, it leads to not the analysis of variance, but the variance of analysis problem. And in using a certain sensitivity method of analysis, it's important to ask this question. Suppose the mortality benefit was even greater than what was observed in the study. Would the pattern mixture result have moved closer to MMRM, or would it have moved away from it? All right. Clearly, you don't want, if there is a mortality benefit, for the pattern mixture method to be ultra conservative. All right. So now I'll move on to the proposal that we are making. So, and this is tied to what the ICH has suggested in their addendum on estimands. And what they say is, under this section on composite variables, that a patient who discontinues treatment because of toxicity may be considered not to have been successfully treated. And the method is based on that. So what we are proposing is prioritizing the reason for missingness and take the time when missing data occurred into account. And by missing data, what we mean is that the patient has dropped out of the study prematurely. So I'm just going to take some examples to show you how this would work, but it's the same idea of making pairwise comparisons. So in this example, there is no missing data. Both patients have data at month 30. Patient B has a bigger decline than patient A, and so we would say that patient A did better than patient B. All right. This next example, only one patient has missing data. Patient B drops out at month 23 because of an adverse event, but there was a big decline for patient A on their 6-minute walk test result. So it's just, it's a matter of deciding what your preferences are. And so one can have a preference that says, even though patient A had a big decline in their 6-minute walk test result, I'm going to say that this patient did better than patient B, because patient B dropped out of the study because of an adverse event, and I think that is a worse outcome. Another example, both patients have missing data because they both had the same adverse event. Again, you would do the comparison at the minimum of their follow-up times. Patient B dropped out before patient A at month 20 as opposed to month 23 for patient A, and you would say patient A did better. Here's another example that they both dropped out because they withdrew consent. Patient B withdrew consent before patient A, but you might say, because they withdrew consent, it's not informative. So I'm going to say this comparison will result in a tie. The next comparison, they have missing data for different reasons. Patient A withdrew consent at month 23. Patient B dropped out at month 20 because of an adverse event. And one would say that patient A did better. Here, patient A dropped out before patient B because patient A withdrew consent at month 20. So the comparison again happens at the minimum of their follow-up times. So at month 20, you can't tell which patient did better, and you would say this would be a tie. With this example, they have missing data again for different reasons. Patient A dies at month 23. Patient B has an adverse event and exits the study at month 20. Comparison takes place at the minimum of their follow-up time, and you would say that patient A did better than patient B. In case 2, patient A dies at month 20. Patient B drops out due to an adverse event at month 23. And so you would say patient B did better than patient A. And the analysis will take place as I had described it previously. So that is the idea behind how this approach would handle missing data. The endpoint becomes a combination endpoint. The rules for handling data are transparent and easily understood. Now, if you don't have missing data, the endpoint just collapses to the primary efficacy endpoint. One thing I'll note is that because this is a non-parametric solution, it can have less power compared with a model-based method. And also the idea of handling missing data can extend to other kinds of endpoints. And this is my final slide before I pass it on to Ron. So for benefit risk, this approach of making pairwise comparisons of hierarchically arranged endpoints, it provides a framework for evaluation of whether benefit exceeds risk, and if so, by how much. And not only that, for drugs that treat the same condition, one can compare their benefit risk ratios. For the missing data problem, this approach, it prioritizes the reason for missingness, and it also incorporates the time when missingness has occurred. And Aaron, if it's okay, I mean, I can maybe take a minute or so to take a few questions before I pass it on to Ron, but if we could also do it toward the end of the talk.
Oh, if we can limit it to 1 or 2 questions, if people have them, I think that's fine.
All right, I'm not hearing any questions. Should I pass it on to Ron now?
That sounds good. Thank you.
All right.
Thank you, Jeetu. So as Jeetu has mentioned, so he has shown basically how to extend the idea of performing pairwise comparisons of patient data based on the set of pre-specified rules to the setting for the evaluation of benefit risk or handling missing data. So during the part of my presentation, I will present a general sample size formula for parallel group trials where the endpoint is the Mann-Whitney U test, which is essentially this pairwise patient comparison idea. So I think everybody should already be familiar with the idea, should be familiar with the Mann-Whitney U test. But I will just give a quick review of the Mann-Whitney U test so everybody on the same page. So it's the Mann-Whitney U test. It's a traditional non-parametric test of the hypothesis, the null hypothesis, that the probability of patient A from the first group doing better than patient B from the second group, this probability is equal to the probability that the patient A is doing worse than patient B. The test statistic is essentially the summation of the outcomes of all the pairwise comparisons. between a patient from the first group and another patient from the second group. Under the null hypothesis, the expected value of this Utest statistic is equal to 0. And the variance of the U is given by the formula here in the absence of tied ranks. When there are ties, the variance formula is a bit more complicated. Then what one would do is one would just calculate the U statistic, subtract the mean from it, and then divide it by the square root of the variance. And the null hypothesis is rejected if this z-score exceeds a certain threshold in the absolute value. So just want to mention that sometimes the formulation here would be if if patient A wins, this is a score of 1. If it's a tie, the pairwise comparison results in a score of 1/2. And if A is losing, then the score is 0. So sometimes you'll see that formulation, but it's just a linear transformation of the formulation here. So they're essentially equivalent. Okay. It seems that even though the Mann-Whitney U test has been around for a long time, but a general sample size formula for clinical trials that use Mann-Whitney U test as the test statistic for performing hypothesis testing, it does not seem to be available. But it is available, only for certain special cases. For example, for a continuous endpoint, there is a sample size formula in the Noether 1987 paper, and also there are sample size formulas when the endpoint is an ordinal categorical endpoint. And the formula, the sample size formula for the ordinal endpoint obviously can be extended to recurrent event endpoints under equal duration of follow-up. So if all the patient had fixed amount of follow-up and there's no loss to follow-up, then you can just compare the number of recurring events that each patient has experienced during the follow-up with each other. And this is essentially an ordinal endpoint, except that the number of categories here in theory can be infinite. So a patient can, in theory, experience infinite number of events during the follow-up period. But a general sample size formula does not seem to be available, and this is what we have worked on. So here, just a setup for our sample size formula. It's a parallel group trial with 2 arms. The null hypothesis is this theta, which is the ratio of the probabilities is equal to 1, because if theta is equal to 1, this is equivalent to saying the probability of A greater than B is equal to probability of A less than B. So that is what the Mann-Whitney U test is testing. The alternative hypothesis is that theta is different from 1. So for example, theta is greater than 1. Then the inputs to our sample size formulas are the allocation ratio. So the percentage of patients in one treatment group versus the other, the theta, the actual ratio under the alternative hypothesis, the significance level, and beta, which is 1 minus power. These parameters are fairly universal to a sample size formula. But what is kind of unique about the sample size formula, in our sample size formula, are these probabilities. And they can be separated into probabilities of transitive relationship under the null hypothesis, and the probabilities intransitive relationships under the null hypothesis, which I'll explain later. I just want to point out one more thing here is that the sample size formula actually will depend on the variance under the null hypothesis, as well as the variance under the alternative hypothesis. However, for our sample size formula, we're just going to approximate the variance under the alternative hypothesis by the variance under the null hypothesis, which is commonly done. But I just want to point that out. So I will now explain what are the transitive relationships and the intransitive relationships. So first I'll explain what is the transitive relationships. So assuming we have 3 patients here, patients A, B, and C, and we have a hierarchy of 2 endpoints, 2 individual endpoints, where the most important endpoint is time to death, and the second most important endpoint is time to the first hospitalization. So if we compare patient A versus patient B, we would say that patient A did better because patient A had a later time to death. If we compare patient B versus patient C, so the patient C is censored at this point. So, so the comparison on the time to death, it's indeterminate. But then if we compare the time to the first hospitalization, we can say that patient B did better because it's a later time. Because the relationship is transitive, which is what we would usually expect, patient A did better than patient B did better than patient C, we would expect that the patient A also, we would say that patient A but also did better than patient C. And that is what happens here. If we compare patient A versus patient C, the comparison on the time to death is a tie, it's indeterminate. But if we compare the time to first hospitalization, patient A had a later to hospitalization. Actually, up to the minimum follow-up time between these 2 patients, patient A did not experience any hospitalization. So this would be a transitive relationship. However, when we do the general pairwise comparison of a pair of patients, intransitive relationships can occur. So this slide shows such an example. So here, if we compare patient A with patient B, again, similarly, patient A would be said to have a better outcome because of a later time to death. Similarly, patient B would have said to have a better outcome because of a later time to first hospitalization. However, now if we compare patient A versus patient C up to the minimum follow-up time between the 2 patients, actually we'll say based on the hierarchy, based on the rules, we will actually now say patient C did better because patient C had a later time to first hospitalization. This is not what you would expect from a transitive relationship. That is, patient A did better than patient B did better than patient C, but now we say patient C did better than patient A. And this is a case where we say this is a severe case of intransitivity. There are other types of intransitivities, which we call moderate and mild, which I'll show you on the next 2 slides. So this is a case where we say this is a moderate case of intransitivity. Patient A did better than patient B because of a later time to death. Patient B is tied with patient C. The comparison up to the minimum follow-up time is indeterminate. But we say patient C did better than patient A. Because patient C did not have any hospitalization during the follow-up, while patient A did have 1 hospitalization. But this is still intransitive because if it was truly transitive, patient A did better than patient B is tied, has tied outcome with patient C, we would probably still say patient A did better than patient C. But the comparison is reversed. And lastly, this is a case of a mild intransitivity. And here we only have a single endpoint, which is a survival endpoint, time to death. If we compare patient A with patient B, this is indeterminate. And if we compare patient B with patient C, this is also indeterminate because patient C is censored early. But if we compare patient A and patient C, we have a clear winner because patient C is a winner because patient C had a later time to death than patient A. So this is also not fully transitive, because if it's fully transitive, if A has a tight outcome with patient B has a tight outcome with patient C, we would expect patient A to have a tight outcome with patient C. But we actually have a winner and a loser here in this pairwise comparison. So in summary, we have three different types of intransitivity: severe, moderate, and mild. As you can see later from the variance formula, I'm not kind of doing this exercise just for the sake of distinguishing between these different types of intransitivity. Actually, they are intransitive to a certain to different degrees, not only because logically they are, with different severity to different degrees, but also their impact on the variance formula. They have different impact on the variance formula, as you'll see later. So this slide, I'll just briefly discuss what conditions are needed that will give rise to these intransitivity. Here, I assume that the comparison of a pairwise of patients is always up to the minimum of their follow-up times, the shorter follow-up time between the two patients. So first of all, we need both conditions in order for the intransitivity to arise. First, patients will need to have different variable duration follow-ups. If all the patients have fixed or equal duration of follow-ups, then there is no intransitivity. So patients will need to have variable length of follow-up. That's the first condition. And the second condition is it will need to be some type of endpoint that's like a survival endpoint, as you can see on the mild example on the previous slide, or recurrent event endpoint, or a hierarchical combination of endpoints, which are the examples that Jeetu has been shown, or some other examples. But if your endpoint is a binary endpoint or a continuous endpoint, actually, even if the patients have a variable length of follow-up with a single measurement of binary endpoint or a single measurement of continuous endpoint, you're still not going to see intransitivity. So both conditions need to occur in order for the intransitivity to occur. So this slide kind of shows the derivation of the null variance. And actually the key observation in our derivation of the null variance is that the null variance involves comparisons of a triplet of randomly selected patients. Because this is the variance under the null, so there's a single population of patients. And with 3 patients, there are 3 pairwise comparisons, and each pairwise comparison has 3 possible outcomes: a win, a loss, or a tie. And because there are 3 pairwise comparisons among the triplet of patients, as a result, there are 3 raised to the 3rd power, or a total of 27 possible scenarios when you write down the possible outcomes comparing 3 patients. Out of the 27 possible scenarios, 13 are transitive scenarios, and the 14 are intransitive scenarios. And some of these scenarios, transitive scenarios, they actually have the equal probabilities because actually up to a permutation, like which patient you now call patient A, which patient you call patient B, and which patient you call patient C, you can actually map the 27 scenarios into 7 distinct probabilities. So there are the 4 transitive relationships, and then there are the 3 intransitive relationships. And these are exactly the severe, the moderate, and the mild intransitive relationships that I've shown on previous slides. So the null variance can be written in terms of these 7 component probabilities, distinct probabilities. And here's what the formula is. And here I'm representing the null variance in terms of the difference between these 2 estimated probabilities. But as you'll see very soon, you can also re-express the variance in terms of the ratio of these 2 probabilities. So little n and little m, they just represent the sample size of each of the treatment group. And the 7 probabilities, P1 through P7, they correspond exactly to these 7 distinct probabilities. So the first 4 probabilities, they're transitive probabilities, and the next 3 probabilities, they're intransitive probabilities. The null variance formula can be written in 2 equivalent ways. In the first way, it's approximately equal to an average of the first 3 transitive probabilities minus 2 intransitive probabilities. In the second expression, which is equivalent to the first expression, you can write the null variance approximately as 1 minus P4, which is the probability of a 3-way tie. So all the pairwise comparison result in the tie. And then a linear combination, a weighted sum of the 3 intransitive probabilities. And as you can see, the coefficient in front of P5, the severe intransitivity is 4. The coefficient in front of the moderate intransitivity is 2. And the coefficient in front of the mild intransitivity is 1. So as you can see, the impact of these transitivity is indeed the severe intransitivity probability has the most impact on the variance formula. So here are just some special cases when applying the null variance formula to special cases. If there's no tie and there's no intransitivity, this is the usual case when we're comparing 2 continuous distributions. So when the outcome is continuous, this is what you have. And in this case, actually all of these probabilities, because there's no intransitive probabilities, these probabilities P5, P6, P7 are equal to 0. And because there's no tie, for example, a continuous outcome, P4, the probability of 3-way tie, that's also equal to 0. Then the variance formula reduces to this expression. If there's no intransitivity, but with ties, so ties are okay, then it's the same expression as above, except now it has a factor of 1 minus P4, where P4 is the probability of 3-way tie. So the more ties you're going to have, actually it will reduce the variance, will reduce the null variance. And for the survival endpoint, oh, sorry, I think I forgot to mention here is that if you have a single survival endpoint, actually that can only lead to mild intransitivity. But with recurrent event endpoints and hierarchical combination endpoints, actually with these type of endpoints, they can lead to any kind of intransitivity, mild, moderate, or severe. So with a single survival endpoint, actually you will not have moderate or severe intransitive outcomes. So so the only probabilities that you need to plug into the null variance formula is just the P4, the 3-way tie, the probability of 3-way tie, and P7, which is the probability of mild intransitivity. As mentioned already, if you're representing your treatment effect in terms of theta hat, which is the ratio of the probabilities, you can also express the null variance of the log theta hat in terms of this null variance of the difference between the 2 estimated probability, just with a factor, which is 4 divided by 1 minus the probability of the 2-way tie squared. So the probability of 2-way tie is you randomly select 2 patients from the null distribution, and what's the probability of having a tied outcome. And this variance formula can be represented as sigma squared divided by capital N, where capital N is the total sample size. So the sample size with both groups combined. And the sigma squared in the numerator, this is independent of the sample size. This is a quantity that is independent of the sample size. With a variance formula, then one can easily arrive at the sample size formula. So in conclusion, the Mann-Whitney U test has been around for a very long time. However, until now, a general sample size formula does not seem to be available. For hierarchically combined endpoint, as Jeetu has mentioned, this is getting more popular, especially in cardiovascular trials, which is essentially a Mann-Whitney U test. The sample sizes are calculated via simulations at the moment for these trials, but which are very time consuming and not straightforward. So what we hope is that now with our sample size formula being available, hopefully the Mann-Whitney U test can gain popularity in clinical trials, because this is actually a good test. It's non-parametric. It does not depend on any distributional assumptions. And this is widely applicable. It does not matter what kind of endpoints or what mixture of endpoints you're going to have. As long as you're able to compare, you can come up with a set of rules to compare a pair of patients. Then the Mann-Whitney U test can be used for your trial to do hypothesis testing, as well as to evaluate the size of your treatment effect. So we hope that with our proposed formula, the Mann-Whitney U test can continue to gain popularity for clinical trials. However, we do want to mention that in order to use our proposed formula, we do now need to build experience around these component probabilities, because we do need to have experience to say, what is the expected probability of a three-way tie, and what are the probabilities of intransitive outcomes in the future trial that we're designing. So some experience are needed. So with that, I think that's the end of my presentation. Any questions for either me or Jeetu?
Hi, this is Ross Boylan. I'm fascinated by sort of a step early on. So the Mann-Whitney test has pairs that it considers, but your whole discussion is about triples. Can you give us any insight into why the triples need to be introduced?
Yes, that's a very good question. Because I think actually the way we look at the null variance in terms of these triplets is our main contribution to the variance formula. Because previously people have only looked at the derivation of these null variances in terms of these pairwise comparisons. But I think by actually looking at these null variances in terms of these probabilities of triplets of patients, actually it illuminates that we have these two distinct broad categories of relationships available. I think previously it was not clear to people that actually like people need to worry about these intransitive relationships. But by looking sort of cast the null variance in light of these probabilities involving triplets of patients, I think it makes very clear that not only we need to worry about the transitive relationship, which is indeed the most common relationships for a problem, but these intransitive relationships actually also have an impact on your sample size calculation. But I would say this is sort of, we just stumbled upon this. It's not planned. We didn't plan to look at these null variance in terms of these probabilities of comparing a triplet of patients. It's just something we stumbled upon.
Yeah, it's not an obvious move, so that's pretty cool.
Thank you. Jeetu, you want to add anything to that?
No, I think you covered it. Maybe I'll just make one brief comment that actually, when you sort of write down the variance, of theta hat using indicator variables, and you sort of work out the math, it basically ends up with a comparison of a triplet of patients. So, and as I mentioned earlier, because we were covering these topics at a high level, it was hard to get into some of these details as well, because we were covering a broad range of topics today. But happy to follow up and give you some more details separately as well.
Any other questions for our speakers?
I have a question. So it's more of a conceptual question. Thanks a lot for your talk. I'm actually very happy to hear that other people are working on how to include benefit risk in clinical trials and how to actually design them to elucidate benefit risk. One thing that I'm tripping up on is that I think your method implicitly assumes a weight. So if you're thinking in terms of how important are different outcomes, then I think implicitly they're somehow as important as they're frequent in your method. Because if I think, for example, let's say we take one outcome that's death and the other, let's just say for really extreme example, treatment burden. So anyone on treatment A would have more treatment burden than treatment B. And then we have some deaths that we observe, and let's say it's like a preventive trial, so death is a really rare outcome. Maybe 1% of patients die. Then I think no matter how good treatment A actually performs, treatment B is going to come out better, because everyone basically will do, almost everyone will do better in treatment B than treatment A by definition. So can you elucidate what's the conditions to apply this method? What needs to hold in it? I think we need to have outcomes that are similar in severity. Is that correct?
So in the sort of list of benefit and safety and efficacy endpoints, you want to include endpoints that are important. Now, to your specific question, those types of situations can arise. But I think, with your example, I think then it's worth asking also the question, how would FDA, when they make sort of that subjective call about whether benefit exceeded risk, how would they go through that thought process, using your example? So, that is not articulated, it's not pre-specified. And to me, sort of the bigger issue is we need to have a framework, for deciding when benefit exceeds risk. What is our threshold for declaring that? And one may have different rules. I mean, to your example, if you feel a different weighting system is better, that is an option. So, the way sort of we presented this approach, just to keep things simple, you could have had a situation where one patient had 10 hospitalizations and died, much later. The other patient had 0 hospitalizations but died earlier. And we would say that the patient with 10 hospitalizations actually did better, right? That's just a rule that is set up to enable a comparison to take place. You can actually modify that rule in a way you think suits your preferences. But I think the key part is that this should be done, we strongly think this should be done in a pre-specified way. And so that it is public, it is transparent to everybody what the criteria were for making that benefit risk assessment. And it does surprise me that, for all these years that we've been doing clinical trials, for efficacy endpoints, we have pre-specified what our risk threshold is for our risk tolerance is for declaring false positives. We set it at 2.5% by reference to a one-sided alpha, but we don't have something like this for benefit risk. So to me, the main push is to move in that direction. And if there are different weighting schemes, people can sort of investigate those. But the main step to me is to move in the direction of finding a way to quantify benefit risk. You can think about, COVID vaccines, for example. Is one vaccine better than the other? I think I see somebody's hand up over here.
Yeah, I had a question for you regarding your artificial example. I wonder if you could bring up that slide easily.
Okay, let me just go to it.
Yes, that one. It looks to me like the first, second, and third were measures of benefit, and the first, second, and fourth were measures of benefit, and the third and fifth were measures of adverse events. So it looks like arm A, let's just say, was more beneficial because the odds ratio was increasing with all of the benefit outcomes. But arm B was safer because the risk was decreasing with both of the AE outcomes. So it does seem that at the end of the day, when you have an odds ratio at the end of the trial, that kind of information would be really valuable to know, that arm A was superior in terms of benefit, but not toxicity or adverse events, because it could then lead to differential selection of who could benefit from this. A more vulnerable patient maybe shouldn't be exposed to this therapy with lots of comorbidities or something like that. So I think in terms of interpreting it, you'd still want to have an idea of how each arm did with respect to benefit and risk, maybe the 4 quadrants.
So I think that might suggest that there could be, one hierarchical arrangement for efficacy endpoints, a separate hierarchical arrangement for safety endpoints. So that is one possibility. And ultimately, a binary decision has to be made as to whether benefit exceeds risk and whether the drug should be approved. So it's with respect to sort of that ultimate question, we had taken this approach.
Yes. No, I think it's a cool approach and a great question to ask. I just think that this might be a piece of the delivery.
Yeah.
Maybe just to add that I think you're right that probably even with this analysis, we'll still want to look at these individual component endpoints.
Yeah.
Too, as a separate analysis. But this analysis that integrates the efficacy and safety endpoints together, it's sort of like when we compare a pair of patients, we're looking at the totality of their efficacy and safety at the same time when we compare the outcome.
Right.
Yeah.
I think another slightly tricky thing is that say death might be seen as both a safety outcome, so anyway, just to make it more complicated, right? But thank you. It's a very nice presentation.
Thank you.
Thank you.
I'll just add separately that, we are very eager to apply these methods on real data sets, and that's been a challenge for us to get our hands on real data sets. But, I think that'll be sort of the real test of how these methods will work. But it is not easy getting access to data from phase 3 clinical trials.
You could ask Dr. Pocock.
That is an option, yes. Yeah, but I think companies will want to know how this is going to look before they will give the complete green light to using it.
Any more questions for our speaker? Okay, well, I hope you join me in thanking both Jeetu and Ron for that fantastic talk, as well as Esti for bringing them into our joint seminar that we've hosted. There'll be a recording available for any of you who want to rewatch any particular segments.
Yeah, it will be posted on the CAPS Methods Core website, but it will take a little bit of time. So don't send me an email tomorrow. But it will be available. And I assume I can get a copy of your slides, Jeetu and Ron?
Indeed.
So I will
Yeah,
post
yeah.
the slides as well. Yeah.
Yeah, and if people have any follow-up questions that occur to them, after this, they're welcome to reach out to us.
Right. Yeah, and thank you for hosting. That was great. Yeah. Okay.
Thank you very much.
Bye-bye. Thank you again.
Thanks a lot for having us.
Yep. Bye.
Thank you. Bye-bye.
Bye-bye.